Thank you very much for the invitation, and I'm delighted to be here. My name is Liang. I'm currently a research scientist at the Cold Cancer Institute of MIT. Today, I'm going to share with you our study on microenvironment-triggered multimodal precision diagnostics. So now in the time of precision medicine, diagnostics are becoming increasingly important in clinical management of cancer patients. And here I'm showing you a typical cancer patient journey. And here one would like to be able to access the risk early, detect the disease as early as possible, diagnose that disease, make an intervention, monitor the treatment, and monitor for reoccurrence. So we know if we can find cancer earlier, we'll have better outcomes. So here's a recent report on number of solid tumors when they are found, when they are localized. The 5-year survival rate is much increased. So today we actually look for most of these cancers by imaging techniques, such as CT scans or pap smears that can be seen under the microscope. And more, there's a new field emerging that is called liquid biopsy. So now we are looking into the bloodstream for shed analytes that come directly from tumors. And these can be metabolites, proteins, exosomes, and more excitingly, tumor-free DNAs. This is an emerging field that is really exciting. The challenge of the liquid biopsy field, however, is that one can only measure in the blood what the cancer shed. So this is a limitation of disease biology because some tumors rarely shed circulating DNAs into the blood, and the blood is a relatively complicated analyte to work on. So what we have been thinking about is whether there are ways to add the molecular analytes to the system, create a signal that never exists in the body, that is engineer a complementary paradigm of the endogenous biomarkers, and we call it synthetic biomarkers. So these synthetic biomarkers are injectable probes, they're ectogenous, and they can be rationally designed to directly interact with diseases in situ, and release diagnostic signals in biofluids with lower background, such as the urine. And they can also be engineered to read by our tools of choice. So this idea has captured the imagination of many in the field, including the National Cancer Institute, who recently hosted a think tank entitled Synthetic Biomarkers for Early Detection to stimulate research in this area. So to design sensitive and specific synthetic biomarkers for cancer detection and monitoring, we leverage the disease hallmarks in the tumor microenvironment proteases to drive the production of the diagnostic signal. And the proteases are a large group of enzymes, and they represent 2% of the human genome, and so far there are over 600 of them identified. And functional-wise, proteases are the key regulators of the extracellular matrix in the tissue microenvironment, and they play a central role in cancer progression, invasion, and metastases. In particular, the proteases MMP9, which is a type 4 collagenase, has been identified as a pan-cancer molecular signature. In breast cancer, MMP9 turns on the angiogenic switch of the tumor when the tumor size are as small as 1 to 2 millimeters. And it has previously been validated to be more sensitive than cell-free DNA in mouse models. So the proteases activity-based synthetic biomarkers are typically made upon a nano-sized core material, such as biocompatible polymers or biologics. And these cores carry multiple strands of labeled peptide substrates. And then the whole sensor can now be injected systemically. They travel in the bloodstream, reach a solid tumor through permeable vasculature, and then these sensors can now access the tumor, and there we design them to be vulnerable to cleave by tumor-associated proteases. And of course, one copy of enzyme can cut many copies of substrates in 1 hour, so now we can get an amplification effect. So when those proteases activity-based nanosensors are acted on the proteases, they release a reporter, and that reporter are made chemically, so they won't degrade in the bloodstream, and then they are able to re-enter the circulation, where the kidney is a 5 nanometer filter. So if these fragments are smaller than that filtration limit, they will be concentrated in the urine. So the way that the whole system works is that one can inject these sensors systemically, it will interrogate the tumor, be catalytically amplified, concentrate in the urine by a size-dependent trafficking, and 1 hour later, one can read the disease signal in the urine. So this is a minimally invasive way to generate molecular analytes by interrogating a tumor in situ. So for cancer detection, the activity-based synthetic biomarkers represent a highly sensitive system, and the limitation in mice is 3 millimeter. The specificity comes from a ligand binding to promote tumor trafficking that I will be talking about in the rest of the talk, or it comes from a multiplexity. Here we can make a cocktail of sensors in addition to just one sensor to be more specific. And in mice which are isogenic and they're all identical genetically, and one can classify the disease with from 3 to 5 different probes. Well, the fun part about this as engineers has been to think about these different clinical use cases that build up off the system, because the synthetic biomarkers is a completely engineered platform, and we are creating these analytes that will be useful to the clinicians. So once you have made a cancer diagnostics, clinicians would like to localize the cancer for follow-on therapy. So here in this study, I wanted to address this clinical actionability gap by combining the imaging capability to our synthetic biomarker platform to visualize the origin of the diagnostic signal. So here I engineered a multimodal probe that can provide both a longitudinal pan-cancer molecular diagnostic and optionally a PET-CT probe for follow-up imaging with the same core material loaded with radioactive imaging tracers. And imaging could then inform clinicians of the site of the primary disease and the degree of metastases. So in this multimodal diagnostics, we wanted to make a single material that can provide both a longitudinal molecular diagnostics through proteases activation and can optionally be turned into a medical imaging PET-CT probe for follow-up tumor visualization. And we call these sensors proteases responsive imaging sensors or PRISMs, and they target tumors through tumor acidosis, which is another ubiquitous environment hallmarks in the range of solid tumors. And they are engineered to be responded to proteases in the microenvironment to release urinary reporters for molecular readout. So we then injected PRISM in a mouse model of colorectal cancer that bears lung tumor nodules. But here the versatile design will allow the sensors to be applied to other tumor types as well, including the breast cancer. And here we performed high-throughput cryofluorescence tomography on whole animals after systemic administration of a fluorophore-film-labeled PRISM, and reconstitution of these cryosections showed the localization of fluorescence signal in the pattern that was perfectly aligned with corresponding anatomical tumors that had performed throughout the lung. And besides fluorescence-based imaging, we further modified the tumor targeting arm of the PRISM sensor and made it optionally carry PET-CT tracers to enable localization of primary and metastatic cancer. So with the characterization of this tumor microenvironment-directed nanosensors, we assayed whether the design could enable multimodal tumor detection in the historically challenging organ for PET and MRI, the lung. So in the typical experimental setting, the PRISM sensors are injected intravenously Urine samples from tumor-bearing and healthy control mice were collected after 1 hour after sensor injection. And then the ELISA readout of Proteases elaborated reporter levels increased as tumor growth processed. And 6 hours after injection, in the same mice that showed significant elevation of the proteases MMP-dependent urine signal, subsequent on-demand loading of the radiotracer copper-64 allowed tumor visualization in PET-CT. So here in the medical imaging tool, we can see a substantially increased positive to negative tumor ratios localized CRC lung tumors. And importantly, we re-imaged the same mice using PET-CT with a conventional tracer used in clinics, which is F18FDG, and there was a significant background signal in the chest cavity due to dramatic uptake of the glucose analog by metabolically active cardiac cells. So therefore, we can see the PRISM sensors here offers a great potential in detecting tumors in the lung compared with a conventional PET tracer. Beyond detection, after treatment, non-invasive monitoring is essential to screen for tumor reoccurrence and assess treatment efficacy. So towards this end, we evaluated the PRISM sensors in monitoring the local activities of tumor in response to drug treatment with 5-FU, which is a standard chemotherapy agent for colorectal cancer patients. So here in the experimental design, we divided cohorts of either lung CRC tumor-bearing mice into experimental groups that received a course of 5-FU treatment and also a control group that received only vehicle. As we expected, the chemo treatment in the colorectal cancer lung disease model significantly inhibited tumor progression compared with a non-treated cohort, which was apparent both in the decreased urine reporter level as well as in the PET imaging readout, even after a single week of inventation. And here on the bottom right, we show that in PET-CT, at the 2-week time point, untreated mice showed substantially increased tumor burden in the lung. So in summary, in this presentation, I've shown you an engineered paradigm wherein a multimodal sensor can be employed longitudinally to non-invasively assess the burden of disease during tumor progression, and as well as responses to chemotherapy. And this modular material platform leveraged generalizable hallmarks of invasive tumors, including the dysregulated proteases activity and the increased extracellular acidosis at the invasive tumor front to enhance the specificity of its diagnostic signal. And for future, this modular platform has the capability to be re-engineered to guide treatment decisions for numerous tumor types, including breast cancer, where it has been really well appreciated that proteases play an important role in disease progression as well. With that, I would like to thank my mentor, Professor Sangeeta Bhatia from the Koch Cancer Institute of MIT, for being a great supervisor and a role model. And I want to thank my collaborators inside and outside the lab. Without their help, I won't be able to show you the study that I presented today. And a special thanks to the Swanson Biotechnological Center, and especially the PET-CT expert, and also my funding resources. I would like to thank you for your attention, and I'll be happy to take any questions from here. And if you have any further ideas to discuss, please feel free to contact me at the email listed on the slides. Thank you.